All right. Everybody with me so far? So instead of one gene determining if a disease or a trait would manifest, many genes would exert a small influence. And none by themselves would cause or explain the disease or trait fully, but together, along with environmental influences, would combine to define an individual outcome. Basically, what we suddenly started to realize that most common diseases, in fact, work this way. So this is basically what started changing about 15 years ago. So now this is also what enabled the very design of genome-wide association studies. So many studies were attempted initially, but very few actually led to meaningful results. Those results would in fact not replicate very well. So a lot of people criticize genome-wide association studies as simply being impossible and unlikely to work. So there's three different things that turned the tide. On one hand, the building of a lot of genomic resources that basically enabled these studies. Number two, a lot of technologies. And number three, a lot of collaboration. Basically, the first element was understanding the genome. Basically, number one, building a reference human genome, and then systematically understanding and cataloging human genetic variation. So basically, building a haplotype map, building the 1,000 Genomes project basically enabled us to now recognize where are these haplotype blocks and how do they vary and how are they inherited. The second part was technological advances. So basically, these microarrays that had millions of DNA variants spotted on them, and the ability to sequence the entire human genome relatively cheaply. And that basically enabled the comparisons of affected and affected families in populations of cases and controls, but also in members of the family with and without the disease. And then the third fundamental change was basically collaboration. A lot of these consortia ended up having participants from dozens of countries across the globe and across every time zone, basically leading to larger and larger cohort sizes. And that was hugely important in sort of changing the tide. So then once you have all of these samples, how do you actually perform a genome-wide association study? So basically, the first thing is technical quality control. So basically removing failed SNPs, removing failed samples, and only maintaining the best ones, because you're looking for a small signal with such a small number of loci and such a small number of cases that have a particular allele at every locus. Small mistakes that are systematic and basically lead to false signals. So that's why you need a lot of technical QC. You also need a lot of genetic QC, basically ensuring that Hardy-Weinberg equilibrium, for example, holds and Mendelian segregations for your trait. You estimate relatedness in your sample, and then you remove individuals who are closely related, because that can actually amplify the signal in any locus that they share. You look for gender biases, and you also remove population structure. As we talked about last time, the first principal components of variation in your sample are largely coming from population structure. And then there's a lot of analysis-based QC. So basically, do some initial rounds of test statistics. Basically, do they show inflation? Do they show biases towards missing data? Do they show specific allele frequencies that indicate problems? And once you have all of that, basically ask how often did I find one allele of the SNP versus the other allele of the SNP in cases versus controls. Okay? And when you see a table like this, what do you notice? You basically say, well, allele G is roughly at the same frequency across these individuals. Let me start closing some applications. I really don't have that much that's in here. So basically, the first thing you'll notice here is that in cases, you see, threefold, change in, so basically, allele A is much more frequent in controls versus allele G appears to be more frequent in cases. This table here. So basically, what you would expect is a uniform ratio between them. Based on these numbers, you would expect to have roughly, 47 individuals for both cases and controls, and roughly 950 individuals for both. So if you just ask how many would I expect for each allele, if this was sampled from the same distribution, if cases and controls were sampled from the same distribution, you would expect to see this. In fact, what you observe is this. And how do you quantify that deviation? Or what you can basically do is calculate a chi-square statistic, which is basically summing up the difference between observed and expected. So, here's the differences, squared divided by expected. Okay? When you do that, you basically have a chi-square, which basically ends up being 24.5. And that has 2 degrees of freedom, so you can see along your chi-square table, where does 2 degrees of freedom with that number fall down, and what is the associated p-value. And then what you end up with is a p-value of 7.3 times. Okay? Basically, this is a very simple test. It's a single marker regression chi-square. And these tests are basically the way to go for most genetic studies. Basically, the association results are based on very arcane statistics. And usually complex multi-marker models are actually less reliable. So what you can do once you have that is basically ask, great, is this my correct p-value? So what's the problem here? The problem is that we're testing millions of these genetic variants. We have millions of SNPs on the array. If we make millions of tests, we have to actually correct for millions of hypotheses that we're actually testing. So the way to do that is to basically say, listen, in linkage, p-value of 0.01, for example, would basically be considered significant because I basically have roughly 50 chromosomal arms that I'm testing. In GWAS, I'm not testing 50 chromosomal arms, I'm testing 1 million independent loci. If you look at just how many haplotype blocks are there in the human genome, you roughly find 1 million tests are basically being performed. If every study has hundreds of these small p-values, this is exactly what you would expect by chance. There's simply no real relationship of the disease. So basically, after a lot of the early studies started not replicating, the community got together and basically said, listen, we're going to define the term genome-wide significance, and we're going to set a p-value associated with that. And this was a very back of the envelope calculation that basically said, if I want to quantify and I have 1 million tests, then 10 to the minus 2 times 10 to the minus 6, that becomes 10 to the minus 8, and then the point, the 5 here is basically how you end up with that. Okay, who's with me so far? So that's where these genome-wide significance thresholds come in. And then this was just a very simple Bonferroni correction. This was a very back of the envelope calculation. But for some reason, it seems to have stood the test of time. And even to this date, we basically have empirical estimates from dense genome-wide maps of common DNA that basically estimate the number to be, roughly this 5 times 10 to the minus 8 out of that was selected, basically 22 years ago now. So Rich and Maringana basically proposed this p-value of 10 to the minus 8 using this back of the envelope. And much later, now that we have a much better understanding of haplotype blocks, it seems to be a good value. So this is what a genome-wide association study actually looks like. So you basically have different p-value cutoffs here, and you have a dot for every SNP in the genome. And the x-axis of your dot basically says what is the genomic position of that SNP. And then the y-axis basically tells you what is the minus log 10 of this chi-square statistic of significance. Okay? And then you end up with these very, very tall things that kind of look like Manhattan skyline. And then that gives a name for this genome-wide association plot to be a Manhattan plot. So you have these tall skyscrapers, and you have these lower height buildings, and so on. And then the big challenge was then, are these results real? Do they replicate? How do we make sense of them? Is everybody comfortable with a genome-wide association study? Again, if you see a report of association from a single study, even with the strictest quality control, there are artifacts that can affect, 1 every 1,000 or 10,000 SNPs, and then escape notice. So strict genome-wide significance is very often not dramatically exceeded in a single study. If you have this single SNP here that appears to, pass your threshold, or this SNP here that appears to pass your other threshold, then, you shouldn't necessarily believe them. Basically, what real hits look like is these very, very strong pillars that are repeatedly found for multiple SNPs in that region that sort of give you reassurance that this is not just a statistical artifact. And usually these true hits are much above your significance threshold. So what you usually do is you replicate your study in a different cohort, in a different ethnicity, in a different study, in a different background, and so on and so forth. Any questions? Yeah. So they basically said, what is the empirical p-value after doing these tests based on, replication studies, based on the number of independent haplotypes, based on the true number of independent tests that you're doing. And that's, there's basically different methods that were used by different groups, but they all arrived at that same conclusion of, this is actually an appropriate correction. I think I lost my pointer. Okay. Any other questions? So this is basically the foundation of these genome-wide association studies. So the chi-square statistic, a p-value for every SNP, a genome-wide association threshold of 10 to the minus 8 or 5 times 10 to the minus 8, and the resulting Manhattan plot. Another way to actually plot these p-values is a QQ plot, where you basically say, what is my expected chi-squared value for every SNP, and what is the observed chi-squared value? And what that basically gives you is a deviation from that that tells you a little bit about, number one, whether your study is actually well calibrated. If you see that the p-values are starting to be inflated from the very beginning, or your chi-square statisticants are being inflated from the very beginning, that is problematic. That means that there's some kind of hidden population structure, that you haven't well calibrated your controls for some kind of artifact. If you see that it sort of follows along together and then starts shooting up, the point where it starts shooting up tells you a little bit about the disease architecture, about simply how many SNPs do we expect to be truly associated with the disease. And then that also allows you to calculate a confidence interval surrounding this null distribution that basically says, well, given some statistical noise, I would expect to be within this range. And if I'm well beyond this range, I know that I have true signal. This also allows you to basically look at subsets of SNPs that are biased, for example, that are in genome-wide enriched genomic annotations, and then see if this QQ plot, in fact, shoots up much earlier, suggesting that you are selecting SNPs that are much more biologically meaningful. And then when you have, this Manhattan plot, you basically can then look within your interval and basically ask, now what is the association of every single individual SNP after I've zoomed in within a particular locus of association. And what you can see here are the recombination hotspots, which basically result in the boundary blocks of these haplotype blocks that are co-inherited. And your association will typically have, a very strong leading point, and then start falling off within that LD block based on, simply the co-association, the co-inheritance patterns of additional SNPs that lead them to be statistically enriched, even if they're not biologically meaningful by themselves. Everybody with me so far? So if you look at Crohn's disease, there were, initially a very small number of hits. Now there are dozens of hits. And those hits were sometimes replicating something that was already known based on linkage analysis, and sometimes putting real diagnostics. And the way to recognize that is that it really depends on the frequency of these alternate alleles. If you look at linkage versus association, these top hit here, NOD2, which was also found by Linkage, had a 5% frequency in the population and an odds ratio of 3. If you have the bad allele of that SNP, then you have a 3-fold higher chance of carrying the disease. By contrast, these IL-23 and these ATG16L1 loci were much lower odds ratios. The odds ratio was, 1/3 or 40% increased, which was basically, you can then ask what is the number of association cases that need to be profiled in order to achieve genome-wide significance, and what is the number of linkage pedigrees that is needed to achieve genome-wide significance. In this particular case, for NOD2, the signal and the frequency were such, the odds ratio and the frequency were such, that you needed about 430 association cases, of which there was a sufficient number in the GWAS study. But for linkage, you needed 1,400 families. Okay. For IL-23R, you can basically estimate using these numbers that you need 800 cases for association, but you need 30,000 pedigrees in order to discover it, given the relatively small odds ratio. And similarly, for ATG16L1, you would need about 40,000 families. So what this basically tells you is that there's a very, very clear reason why some of these GWAS hits are not discovered by linkage, because we simply have not profiled that many pedigrees. And it also tells you about these different regimes where these work best. If you have strong odds ratios, then linkage is actually very powerful. But if you have weak ones, then genome-wide association studies are very powerful. And then you should also realize that the frequency matters greatly for the success of genome-wide association studies. If the frequency of this variant was lower, then you would need relatively few families to associate it, to discover it, but you would need a huge number of GWAS cases. Any questions so far? Great. So again, combining studies yields much greater power. So when you have a discovery cohort and a replication cohort, you can basically combine them together to basically carry out meta-analysis and then achieve much greater power. And then a lot of the progress in GWAS has been the result of multiple meta-analysis. So here's one example where you basically have the progression for Crohn's disease of how many genes are discovered. So here I showed you some of the early work where you have only a handful of genome-wide significance hits. And then as you start carrying out more and more meta-analysis, you basically end up with this dramatic increase in the number of hits. So as you have larger and larger studies, you don't simply get this continued linear increase in the number of hits, but you get an exponential. The same inflection point was found in many different GWAS studies. So basically, if you look at schizophrenia, as you increase the number of cases, you basically include more and more collaborators, and then the number of cases increases, and then suddenly you start discovering more and more loci. So with 10,000 cases, there were only 5 loci that were discoverable. With 3,500 cases, there was not a single one that was discovered. With 10,000, you suddenly had 5 that were barely passing this genome-wide significance. With 35,000 cases, you now had 62 loci. And right now, with 65,000 cases, we're reaching 265 loci for schizophrenia. So schizophrenia used to be thought to be, not due to common variants. And then suddenly you basically realize that this was just a question of power. That basically as you increase more and more the number of cases, you suddenly had the ability to discover it. So you, of course, do microarrays still to this date. Microarrays are dramatically cheaper. The problem with whole genome sequencing is that the number of hypotheses goes off the roof. You no longer have 6 million tests. You suddenly start having, I don't know, 1 billion tests based on the number of variants that are actually being ascertained. So We're going to talk a little bit about sort of rare variant analysis, but genome-wide rare variant analysis is still dramatically underpowered. You need many, many more cases. Correct. Basically, the macroarray only tests a fixed number of common SNPs. And then you impute those SNPs, as we showed in the last lecture, and then you carry out those association with imputed SNPs. Any other? Yeah. Correct. Yeah, your power is a direct consequence of your sample size. And this was not true just for schizophrenia. It was not just true for Crohn's disease. For many different studies, as you basically cross that threshold, and that threshold was in fact different for different studies. So for Crohn's disease, the unit was 1,000. So you basically looked at 2,000 individuals, 4,000 individuals, 5,000 individuals. For schizophrenia, the unit was 3,000. 3,000, 6,000, 18,000, and so on and so forth. For height, similarly, times 2, times 3, times 9, times 18. And the number of loci was basically very, very few to none, and then suddenly a huge number. And this is actually completely expected if you assume that evolutionary selection is maintaining these variants at low frequency. How? You basically, any of these variants, basically genetic variants are bombarding the genome all the time. And some of these variants have, a distribution of effects. Basically, all of these variants have a distribution of effects. As these variants that are, entering the population reach an effect that is, too strong for evolutionary selection to be tolerated, then they're going to be maintained at low frequency. So all these variants are going to be basically, leading up to that frequency just by random genetic drift, and then stopping at that frequency. So as you increase the sample size and you get to that frequency where selection hits, you're basically, crossing that threshold, and you start you need to increase your power because that distribution, being continuous after that. But you can sort of imagine this threshold that selection will start acting at. And in order to get at that threshold, you need your discovery power to be correspondingly large. Okay, so this is at least charging, and I am on the correct network. So do you still see me over there? Okay. So we found the same story in type 1 diabetes, type 2 diabetes, and serum cholesterol levels in every common chronic disease. So larger samples basically lead to a lot more discovery, and they also lead to new biological insights. For example, with schizophrenia, it suddenly became clear that it was a heritable medical disorder, and its genetic architecture was in fact similar to non-brain diseases and traits. Many genes also enable the recognition of key pathways and processes. So, for example, there's a lot of enrichment in, calcium channels and fragile eggs and neurons and specific enhancers, which would not simply be possible to recognize using few loci, but becomes possible to recognize using many, many loci. Okay. Who's with me so far? Awesome. So as you start doing this across many, many disorders, you can basically start populating this GWAS catalog that was initially started by NHGRI, the National Human Genome Research Institute. You can basically sort of color every association based on the trait that it is associated with, and then start painting the genome. And you saw that graph initially where there was only like 10 or 15 or maybe 30 in the early 2000s. By 2011, there were 1,300 published GWAS signals at this threshold across 221 traits. This is what it looks like today. So this is just a humongous success of genome-wide association studies. We basically have 70,000 associations across 5,000 studies, and 3,300 papers, and across just a huge diversity of traits. Look at digestive system diseases, cardiovascular, metabolic, immune, nervous system, liver, lipid, inflammatory, hematological, body weight, cardiovascular, all kinds of other measurements, drug response, biological processes, cancer, other diseases, and other traits. You can basically see this incredibly dense map. So great success. So what's the next challenge? Well, making sense of all that. So basically, this is a huge success in discovery of genetic bases, but a huge failure in terms of discovery of mechanistic bases. And the discrepancy between them is a humongous opportunity for you guys. Basically, you are at a generation where there are, 70,000 puzzles to be solved. So any one of them could actually lead to a new drug that can be completely revolutionary. It can be $1 billion business. And it's up to you guys to now use the tools that you've discovered and that you've learned about in this class to just go at it and just understand how these loci are working and, start paving the path for therapeutics. Okay? No pressure, but the charge for your generation. So how do we deal with interpretation? So basically, on one hand, you can work with individual loci. On the other hand, you can start understanding global signals at the system level. So, the goal of GWAS was to inform the biology of the disease in an actionable fashion. So why is it an actionable fashion? Because you know that that particular locus, which happens to have escaped selection, if you change a single nucleotide in that locus, you will have a detectable, reliable, and statistically significant change in the corresponding disease phenotype. So that basically means that there's a direct causality, that basically, despite all of the feedback loops that exist in your genome and in your cellular, machinery, these feedback loops do not interfere in that particular pathway. You can basically intervene here and have an effect all the way to phenotype. That's a big deal. That's basically causality. And that basically means that if I intervene genetically all the way down to the DNA level, and of course, transcriptionally, epigenetically, at the protein level and so on and so forth, I will have an effect at the organism level. Is that clear? Why genetics is just so darn important? Whereas if you look at model organisms, great, you perturb that gene in a mouse, that mouse has a dramatic phenotype, but perhaps the feedback mechanisms in human are different, and you will actually not have the same phenotypic consequence of perturbing that in a human. Whereas right now, because we have genetics, we actually have the advantage of knowing that it will have an impact at the disease level. But there's a huge challenge of interpretation. Basically, most associations do not identify specific genes. They do not identify causal mutations. They only give you pointers to regions of the genome with causal influences on the disease. And in order to develop and act on a therapeutic hypothesis, you must go much further. You must understand what gene is connected to the disease. You must understand what biological process is implicated through these genes. You must understand the cellular context, what tissue, what cell type, when in life, at what stage of development, in the context of what environmental stimulus does that process become relevant to disease? And what are the specific functional alleles that actually perturb the process and promote or protect from the disease? So, this is what we're going to discuss now. How do we use the tools that we learned about from epigenomics, regulatory genomics, and so on and so forth to actually shed light into these variants? Again, the promise of these genetic studies is that they can give us disease mechanism, new target genes, new therapeutics, ultimately personalized medicine. But the challenge is interpretation. And if you start looking at these genetic loci of association, in this particular case, I'm showing you body mass index. This is a measure of obesity. The strongest association for obesity was discovered in 2007, and it became a huge focus of intense study. More than $1 billion have been spent across industry and academia to understand the function of these locus and these genes. and to develop, therapeutic trials based on targeting FTO and so on and so forth. Nothing came out for more than 10 years. So why is that? If you look at the locus of association, here we are. It sits right smack in the middle of the first intron and the beginning of the second intron of the FTO gene. There are 89 common variants, but none of these common variants is in fact perturbing the protein of the FTO gene. None of them is exonic. None of them even disrupts an amino acid or a codon. So what that means is that we need to actually understand the non-coding circuitry of this locus. And this is not just an exception. This is true for 93% of GWAS. In 93% of cases, when you open up the hood, you basically realize there's simply no protein coding alteration. Who's with me so far? Raise your hand. Awesome. So that means that we don't know the target gene because, it could just simply not be FTO. It could be some other gene nearby. And in this particular case, it is actually a gene that sits 1.2 million nucleotides that way. I mean, that's basically, a building over in this resolution. It's very, very far away. And, the theory doesn't have the causal variant not known. How do we figure out which of these, 89 variants is actually the causal one? The cell type where it acts is not known. If you had a gene, you might be able to say, well, these are the cell type where the gene is expressed. But if you don't even have the gene, how do you know what cell type something is acting in? And then, of course, not knowing the gene or the cell type or the causal variant, you don't know the relevant pathways, and you don't know the resulting mechanisms. So how do we address that? Well, that's where all of the stuff that we've learned so far comes in. So basically, the ENCODE project that we heard about, the Roadmap Epigenomics project that we heard about, they're basically focused on annotating the non-coding genome, linking non-coding elements to their upstream regulators and to their downstream target genes, elucidating the intermediate molecular and ultimately cellular phenotypes that result from these genetic variants, so that we can get at all those deliverables of finding the right cell type, the target gene, the causal variant, the upstream regulator, the relevant pathways, and the intermediate phenotypes. So epigenomic studies have basically been carried out across hundreds of tissues and cell types, across dozens of different epigenomic maps. We can actually start putting all that together to map chromatin states across hundreds of tissues, to group enhancers into modules of common function, to recognize motifs associated with these modules, and to start linking together these regulatory regions to their target genes. The ultimate goal is to have for every one of these regions exactly this kind of diagram, which basically tells you what are all of the causal variants or the putatively causal variants within this region, what are the associated variants, what are all of the enhancers and regulatory regions that these genetic variants are found in, what are all of the cell types where these enhancers become active in, what are all of the regulators and their motifs that are associated with these regulatory regions, and lastly, what are the target genes that are predicted to be targeted by that. Can everybody see this? So basically, if you knew that, you could do a lot more in the prediction of mechanisms. So we basically built tools for doing that systematically. So we've been part of the ENCODE project, of the Roadmap Epigenomics project, and we've basically started systematically annotating these non-coding variants. So we've developed a tool called HaploReg with Luke Ward that basically allows you to figure out for every genetic variant what are all of the genetic variants that it is co-inherited with, and then what are all of the evolutionary conservation and epigenomic and regulatory motif and linking information that allows you to predict where it may be acting and how. And you can use this information to now start getting a lot of systematic insights into disease mechanisms. So the first thing that you can do is basically ask, let's use all of the genetic variants jointly. Basically look for epigenomic annotations in specific cell types that are enriched in these variants. So if you take genetic variants associated with height or type 1 diabetes or blood pressure or cholesterol, and you start overlaying the genomic regions that are active in different cell types. You can basically ask which regions are active in stem cells or immune cells or heart cells or liver cells. And you can ask, are these classes of epigenomic annotations enriched in genetic variants that are associated with specific traits? We're basically bringing genetic information and epigenomic information and correlating the two. And the first thing you will notice is that different regions and different traits are enriched for specific cell types. And it almost paints a diagonal here. This is a cartoon, but this is actually a subset of a real matrix. I'm going to show you in the next slide. Is everybody with me here about the principle of what we're trying to achieve? We're basically trying to say, are we recurrently hitting epigenomic regions that are active in specific cell types in order to then gain insights as to what are the cell types that are underlying the disease? Raise your hand if you're with me so far. Awesome. So this is the real matrix from which I pulled away these data. So you can see here, genetic variants associated with embryonic stem cells. I'm going to do something that I might regret. So genetic variants associated with height are enriched only in one cell type here, and this is embryonic stem cells. Genetic variants associated with self-reported allergy or type 1 diabetes or celiac disease or multiple sclerosis are all associated with T cells and B cells and generally immune cells. If you look at genetic variants associated with blood pressure, you basically find no enrichment whatsoever across all of these different cell types, except for enhancers active in left ventricle. This is the place where the blood pressure gets built up before the blood is pumped in the rest of the body. If you look at cholesterol, you basically find these very, very strong enrichment consistently across all these different measures of cholesterol for genetic variants that overlap regions that are active in liver. Again, this makes a lot of sense. If you look at fasting glucose-related traits, this is the basis of type 2 diabetes. And what you find is, again, the strongest enrichment it found for pancreatic islets. It makes a lot of sense because, again, the immune system is attacking the pancreatic beta cells in these islets and then changing the ability to sense insulin, and that's what leads to changes in fasting glucose levels. If you look at inflammatory bowel disease, you basically see this very strong enrichment for immune cells, which makes a lot of sense given the inflammatory component of bowel disease. And if you look here, you basically see this dramatic enrichment for digestive tissues, which again makes a lot of sense because it seems to be an interplay between non-coding elements acting in bowel tissues and non-coding elements acting in immune cell types. If you look at Alzheimer's disease, you don't see an enrichment for brain. And that's a problem, right? Because you would expect Alzheimer's to act in brain. But in fact, what you're seeing is a huge enrichment for monocytes, and specifically CD14 plus monocytes. What does that mean? That basically means that there appears to be some kind of immune component to Alzheimer's disease. Why do you not have the majority of brain cell types are in fact excitatory nerves and inhibitory nerves? And then you have oligodendrocytes and you have astrocytes, and all together they make up maybe 90% of the cells in the brain. And this is profiling bulk tissue at the genomic level. By contrast, microglia, which are the resident immune cells of the brain, and in fact are marked by CD14 plus, the same cell type or very similar cell type to monocytes, microglia make up less than 10% of the brain cell types. So that basically means that, yes, there is an association with brain, and that association appears to be specifically with the immune cells of the brain rather than with the neurons. Who's with me so far? Cool. Isn't this kind of weird, right? That we can basically, in a completely unbiased fashion, look at many, many different traits and many, many different cell types and start painting maps and, not only recapitulating decades of biology to recognize, for example, that multiple sclerosis is an immune trait rather than some kind of neuronal trait. And that, these very, very subtle associations with fasting glucose and, pancreatic islets. And, this thing just jumps right at you. This is kind of cool. And then we go at it. And we basically, wrote this paper together saying conserved epigenomic signals in mice and humans reveal immune bases of Alzheimer's disease. This was, a big turning point in the study of Alzheimer's, where suddenly a lot of the focus, if you look at where is the pharmaceutical industry going and where is the research going, a huge focus appears to be specifically in these immune cells, in these microglia. So how did we address that? We basically looked at early Alzheimer's and late Alzheimer's in a mouse model. And we looked at epigenomic changes that happen early and epigenomic changes that happen late. And we found that indeed there's a repression of neuronal processes and an activation of immune processes. But the activation of immune process appears to be happening very early in the disease, whereas the repression of immune process of neuronal process appears to be happening late. So a view that was traditionally thought was that maybe all of this neuronal loss is in fact leading to inflammation. In fact, this study sort of flips this around and says, well, maybe inflammation is the causal component rather than just simply a consequence of neuronal loss. So we can use that to basically start linking all of these traits to specific cell types and clustering all these traits together. And you can see here how, for example, Alzheimer's disease, is not clustering with the brain; it's clustering here with monocytes and immune processes. So that was basically looking globally at enrichments and gaining insight into sort of global patterns of the disease. But we'd like to also use these techniques that we developed now go back into individual loci. And in this particular case, this work was led by Melina Klausnitzer, who was a visiting scholar in my lab, who basically said, hey, can we use these techniques to actually start understanding these locus that I showed you earlier in this FTO region? So the goal is to identify what are the cell types where it acts, what is the causal SNP, what is the upstream regulator, what are the targets, what are the processes, and then, start intervening to change that process. So here are the things that we would like to do. We'd like to start with a region of associations shown here in pink. From all of these genetic variants, figure out which one or which ones are the causal ones that are responsible for the association. What are the tissues and cell types where this region is acting? What are the target genes? What are the cellular phenotypes and the organismal phenotypes downstream of those target genes? And who are the upstream regulators that are binding these region and whose binding changes because of the nucleotide? And we basically went after all of that and applied it to the FTO locus. Here's the locus again, no protein coding variants. And there was a lot of hypothesis that it may be a regulatory role, but both the target gene and the tissue of action was a matter of great controversy. So we basically asked, well, what do the chromatin state maps that we developed, that we learned about in the epigenomics lecture, what do those maps tell us? And what they found was this 12 kb super enhancer, this massive, massive enhancer that spanned 12,000 nucleotides in the first half of these locus. And this region was not active in brain as many people had hypothesized. Many people thought that this FTO locus might be acting to control your appetite, your choice of foods, or whether you're going to go to the gym or lay down. And in fact, this was not acting in the brain. It was acting specifically in the fat tissue, in specifically pre-adipocytes. These are the cells that give rise to your white fat cells and your beige fat cells. So white fat cells are the cells that store lipids. When you eat too much, the excess calories are basically stored by your body. And that used to be a very, very helpful thing because we would go through periods of starvation and then periods of, feasting. And during the periods of feasting, it was actually very beneficial to be able to store store all these excess calories as high energy molecules known as lipids that you would then store away and then use when you were in periods of starvation. And that's what white adipocytes do. They basically store all this energy. They're your best friends if food is scarce, but they become your worst enemies if food is overabundant. Beige adipocytes are very helpful. They basically help you heat up. If you're in a cold zone, your brain basically sends a stimulus to your body, and there's also brain-independent stimuli that basically make your adipocytes burn these calories to heat yourself up. How do they do that? They basically depolarize the mitochondrial membrane, and they lead to transfer of protons, change in the polarization, the depolarization basically of that membrane, and then a loss of energy as heat. And all of that heat basically makes you feel warmer, and that's great. That's a very helpful process if you're trying to heat up, and it's also a very helpful process if you're trying to burn calories down. And this region appears to be acting in the precursor that gives rise to the decision as to whether to become a fat storing cell or a fat burning cell. What about the target genes? So in this particular case, FTO, as I showed you earlier, is basically right smack in the middle of that genetic association. There was no other gene nearby. And if you look at a lot of the interpretation of GWAS, basically many people are saying, oh gosh, GWAS is hard to interpret. Why? Because sometimes when I look at my genetic locus, there are 5 genes in my genetic locus. In this particular case, there's only 1 gene in your genetic locus. But what genes are in your genetic locus has nothing to do with the disease. What matters is what genes are in the regulatory locus, not the genetic locus. The genetic locus simply tells you about the transmission of that genetic variant in order to find what is the causal variant. Once you have the causal variant, of course, you look within that region to find the causal variant. You look within your haplotype log to find the causal variant. But once you have the causal variant, you have to understand what does that causal variant do. And that's where you have to understand the regulatory domain. And you remember the high C lecture where we basically looked at this 3-dimensional conformation of the genome. In this particular case, when you look at 3-dimensional conformation of that locus, you basically found this dramatically large locus on one side of the 3-dimensional conformation domain, this topologically associated domain, on one side of the genetic locus and on the other side of the genetic locus. So any of these genes might actually be the true targets. What Melina did is gather both homozygous risk and homozygous non-risk individuals, took out adipocytes from these people, and then measured the level of expression for all of these genes nearby. And what she found was dramatic. She basically found that 2 genes showed a 2-fold increase in expression for the risk individuals. And that 2-fold increase in expression was found for IRX3 and IRX5. IRX3 sits 500,000 nucleotides away from FTO. IRX5 sits 1.2 million nucleotides away from FTO. So this is a very, very long range interaction. The other thing you'll notice here is that this is a gain of function. So most of the time we're thinking, oh, what is broken by the disease variant? In this particular case, this was overactive by the disease variant. What was broken was the brakes, and therefore these were overactive. And the third surprise was that this increased expression was specifically found in pre-adipocytes. If you looked at whole adipose tissue, there was no change in expression of these genes. These genes increased in expression early in the developmental path of pre-adipocytes on the way to become either beige or white adipocytes. But then, once that decision is made, these genes turn off. It was extremely important to basically measure the specific developmental stage and the specific cell type and the specific issue. Otherwise, this effect would have been undiscoverable. So then we did a lot of work in actually finding the causal variant and the upstream regulator. To basically find the upstream regulator, we basically looked for the expression of the corresponding factors, and to find the causal variant, we looked at the conservation of individual regulatory motifs. And what Melina noted here was that the T2C alteration found in the risk individuals was disrupting this AT-rich motif this AT-rich interacting domain protein, ARID5B, and was basically causing loss of binding of ARID5B. ARID5B normally binds this motif and acts as a repressor of the super enhancer. And when ARID5B no longer binds, this enhancer is de-repressed and RX3 and RX5 are overexpressed. And we basically did conditional analysis of disrupting the cis and the trans of that locus. And what we found is that you needed to have both the cis and the trans intact. You need to have both a working motif and a working protein in order to see successful repression of the enhancer, in order to see successful repression of either of the two target genes. Before the transacting repressor, these regions became de-repressed and overactive, and these genes became de-repressed and overactive. Everybody with me so far? So we basically have the complete circuitry. We have the upstream regulator, we have the causal variant, we have the tissue, and we have the target genes, right? But I still haven't told you anything about obesity and how this could be acting. So let's look at what are these genes actually doing. Okay? So what Melina did was a correlation analysis to basically look at the expression of IRX3 and RX5 in relationship with every other gene in the genome across 20 individuals. And what she found was that IRX3 and RX5, both of these target genes, were positively correlated with lipid metabolism and negatively correlated with mitochondrial function. And that basically suggested that as the genes become de-repressed, you're basically switching away from mitochondria and into lipid metabolism. This was in fact reflected in the cellular phenotypes of these genes. The adipocytes were much larger and the mitochondria were much scarcer. So she basically had a complete circuitry here of the upstream regulator, the driver nucleotide, the downstream target genes, and then the processes of deciding whether you're going to burn energy or store energy, thermogenesis or adipogenesis. And that basically allowed us to now start manipulating each of these targets in that circuitry to switch between these phenotypes. The result was basically dramatic. We found that at any one of these stages, we could actually flip back and forth between lean and obese phenotypes. If you basically changed the expression of IRX3, overexpress it or to knock it down, you basically found that in the risk individuals, you would lead to a massive increase, and in the non-risk individual, you would lead to a massive decrease in relative oxygen consumption rate, which is basically a measure of thermogenesis. If you go to mice, you basically found that you could down-regulate this gene that becomes overexpressed, IRX3, and basically the mice would lose 50% of their body weight. If you put these mice in a high-fat diet, normal mice would gain weight. The IRX3 dominant negative mice would simply be unable to gain weight. You would feed them all the fat you want, you would not be able to get them to gain weight. And these mice did not change their appetite or their exercise. What they did is simply burn more calories when they're awake. You can see here when they're awake, they're burning more calories. And when they're asleep, they're burning more calories. So these mice were just simply thermogenizing. And then to prove that this was due to that single nucleotide variant, we basically went ahead and edited that single nucleotide variant. We went from the protective allele to the risk allele and back to the protective allele. And what we found is that changing that single nucleotide, a million nucleotides away from these two genes, was basically leading to a massive overexpression of these genes if we go from the non-risk to the risk. And if we edit that nucleotide back, massive decrease in expression. And if you look at the cellular phenotypes, that single nucleotide change out of 3.2 billion nucleotides was basically able to take primary adipocytes from risk individuals who were completely unable to thermogenize and completely restore the process of thermogenesis. So that basically established that causality that we're after. So what that basically says is that we now have a mechanism, which is a completely new mechanism, for controlling obesity. Instead of changing your exercise or changing your appetite, you basically change your metabolism. You can basically shift between a white adipocyte state of lipogenesis or a beige adipocyte state of thermogenesis. just because of that single nucleotide in this exome. So this is what we have to do for the remaining 70,000 associations. Sounds good? Who's up for it? All right, so this is what I told you about today. So we're going to stop here. Basically, we talked about Mendelian inheritance. We talked about genome-wide association studies. We talked about linkage in a simple trait and polygenicity in complex traits. We talked about how to carry out a genome-wide association study. What are the statistical tests, and how do we recognize, true associations? And how do we then go about interpreting these regions, both globally in terms of the enrichment of these loci, as well as by detailed analysis of the circuitry of a single locus. All right, thanks a lot.